So take a couple minutes. These are totally fake. I just made this up. But there's a consensus sequence buried in here somewhere. So you do the same thing. I've lined up all of these promoters, supposedly, from different species, promoters of the same gene, say. So see if you can find if there's an average DNA sequence in here somewhere. Make the consensus. Take about 1 minute to do this. If you end early, make sure you've completed the Socrative quiz. Raise your hand if you have any questions. It's awfully quiet. What about the first column? Is there a consensus for the first column of nucleotides?
In
A? I'd say no. You're looking for, in every column, the most frequent nucleotide, but especially if it's mostly one nucleotide. So in column A, I wouldn't conclude that there's any particular agreement or consensus.
Well, there's more A's than others.
Yeah, but there are not that many A's.
So,
3 out of 10? 3 out of 8? Not enough.
okay, so not every column's gonna have one.
Yeah, not every column is going to necessarily have a consensus nucleotide. Somebody found one yet?
No, it's a question.
Yeah, please.
I realized that in the video, what you put the most was T and A's on the 35 and the 10 findings.
Yeah.
And well, that also counts as a consensus sequence here. Is there like a reason we learned before that normally the place where the RNA polymerase binds is full of G's and C's, and is it just a coincidence that there were a lot of T's and C's?
Basically, yeah. It doesn't, yeah, the reason
So it could be like G's and C's.
that there are more A's and T's in the minus 35 and minus 10 sequences seems to be a coincidence. Yeah.
In column 7, I think it's 7.
Let's see. 1, 2, 3, 4, 5, 6, 7, 9, 11, 13, 15, 17. Okay, so column 7.
Yeah.
8 and 9.
So in column 7, we've got 1, 2, 3, 4, 5, 6 out of 8 A's. And then there's a bunch of C's in the column next to it. What next?
Bunch of A's in the column next to them.
A's, 6 T's in that column, and then we go back to, there's 3 G's. That's not, we're looking, say, for more than 50%. That's not a true genetic threshold or cutoff, but for this purpose, let's say 50% or more. So it looks like if there's a consensus sequence in these promoters, that would be it. And that's how biologists would say, in general, proteins that bind this promoter sequence are going to recognize the sequence a cat. One of the longest sentences you can create using the 4 letters in the nucleotide code. Okay. So that's the concept of a consensus sequence and how scientists develop them. It's a figment of our imagination. They're from real data, but the concept of the consensus sequence is it's an average. Yeah.
So what you did was you took the most occurring one.
Correct. So in this column of nucleotides, there are more A's than anything else, and at least 50% of them are A's. So it seems like, on average, when a protein binds this promoter sequence, it's recognizing something specific about each of these sequences. Did any of those sequences actually have the consensus?
You mean all of them had the same
Oh,
one?
yeah.
Yes, exactly. Was the consensus sequence actually a real sequence that existed in one of these species? Just one. I totally didn't do this on purpose, but honestly. But it turned out that the cat was the one where a cat was part of the DNA sequence. So again, the point is that proteins have some give to them. Just because a protein is specific to binding a particular gene's promoter doesn't mean it has to bind to the consensus sequence. It'll bind something that's sort of like the consensus sequence because the consensus sequence is an average. It's just a useful way for geneticists to say, in general, proteins that regulate this gene will recognize a sequence that looks something like this, ACAT. Even, yeah.
Is there a guideline for the number of repeats that would make it, say, more recognizable by standard? Like they want a certain number of repeats? Because, I mean, that's a relatively short sequence that, I mean, could be unspec to anything.
So a couple of points. Repeat sequence not necessary for binding of transcription factors. So transcription factors don't necessarily bind the same sequence over and over again. But it's an interesting point that this is only a 4 nucleotide sequence. And we've looked, I think we've done the math before, how frequently 4 nucleotides in a row will show up in the genome. So how frequently do you expect to find a 4 nucleotide sequence? Probability of getting an A at the first position is 1 in 4. So the probability of getting those 4 specific nucleotides in a row is 1/4 to the 4th power. Oops. I don't know what that 6 is doing there, forget about that. Yeah, so 1 in 256 nucleotides. Which must raise a question in your heads, I hope. Wait a second, if things like TATA binding protein, eukaryotes now, that recognize the sequence TA, that's going to be every 256 base pairs in our billions of base pairs in our genomes. So TBP then, these transcription factors or sigma factors in prokaryotes, they're not the only thing that controls transcription. Because if they were, if all you needed to know was 4 bases in a row, like a cat, to know that's where a protein is going to bind to initiate transcription, then everything in the genome would be transcribed, almost. Genes would be everywhere, which isn't true. So there are other proteins that are involved as well.